I acknowledge that we meet today for this seminar on beautiful Wurundjeri country. The Wurundjeri people have cared for the land for thousands of years as part of the Kulin Nation, and pay my respects to elders past and present, and also any Indigenous people and elders that may be in the room. It's an absolute pleasure to introduce Edwin Hawkins today. Edwin actually did his PhD here at wehi with Phil Hodgkin in the Division of Immunology, where he pioneered the use of long-term live time-lapse microscopy for the analysis of cell fate tracking in vitro. And after he was awarded an NHMRC Early Career Fellowship, he went to the Peter Mac, which was at the time in East Melbourne, where he studied the molecular mechanisms that regulate lymphocyte fate, and he did this with Sarah Russell. And at this point, he wanted to expand his studies to really go in vitro and pioneer the use of 2-photon microscopy to study immune cells with a particular focus on leukaemia. So he relocated to Imperial College in London, where he received fellowships from the Wellcome Trust and the European Haematology Association, and established 2-photon microscopy techniques, which led to his beautiful science paper earlier this year. He is now a lab head in the Immunology division, and he's applying these techniques to study immune responses and function of blood cells in the bone marrow, and we're going to hear about this today. So thank you, Edwin.
It's on, everyone can hear me? Okay, thank you all for turning up today. So I'm going to start by introducing a problem that's very familiar to anybody here in the audience that's been doing science for long enough, probably maybe not so many of the honours students, depending on how good you are already or how many things have failed. But the common problem we have is we start off with a population of starting cells, and then we do something to it and measure the end output. So this might be we knock out a gene of some sort, or we change stimulation. And this is brilliant. It's worked for very many years, and there's lots of ways that we can take the readout of what happens at the end. So that might be by doing a Western blot, where we've got two different conditions, and we look how that protein changes. Or if anybody who sat through numerous immunology talks has ever done, is that's looking at it by flow cytometry, where we look at a number of markers and then compare the whether populations are there or aren't there. So this technique works very well. The problem about it is we throw away a lot of information about the pathways that the cells went through to get to that. And this is just a typical graph that would be something shown by Tim Schroeder is, all right, well, what happened to all of these individual founders that we put in culture at the beginning of this experiment? There are so many different permutations of what could have happened to give us this end event here of this population of cells. So we could have some that went through a few divisions. We could have one where it produced a daughter cell that then did not a lot, but made the end cell. We can have situations with death, or we could have had one daughter cell give us the predominant readout there. So we're still very uncertain about what could have happened. Also, you can then complicate this a little bit further as you can do what are the classic experiments of those done with division tracking diets. And that's shown here for CFSE. And what happens there is we label a population of cells with this dye, and then they dilute it out as they go through divisions. So now we have information about, well, what is going on with that progeny, those progeny, and how they're separated into these different divisions. But we still don't know what happened to each of those founder cells and who's actually contributing to that end response that we're measuring. So again, did we have one cell here at the beginning divide to give us something that gave us all of these dividing progeny at the end, and then the other one didn't do very much at all? Or is it very much a process where we get the same fate for all of them? So what we do a lot now in immunology and leukemia studies is to look at all these processes at the same time. So we might look at how we're getting cell death at the same time in these populations of cells, how they then differentiate to give us one fate, now doing this again by flow cytometry, how they give us another fate at the same time, and then how they might actually give us both of these as well. But again, it's really, really hard for us to know actually how these cells were generated and where they came from. So the process that I developed a long time ago, which a lot of people were interested in as well, was, well, let's take these individual cells and let's put them in culture. So these are the cells just as you saw before, and then let's videotape them. And that's really, really good, because now instead of knowing information about, well, what was our beginning population and what was our end population and how they look, we can now go through and track and manually look at all these cells and see who did what. And that was a remarkably successful experiment, but it was remarkably disastrous, and I talk about this all the time, because to analyze this, you have to do it all manually. And it took us about 3 to 6 months to acquire the data, about 1.5 years to analyze the data. And because I was the genius that decided that the experiment was going to be like this, then I was the genius that then had to analyze it. So you can imagine it's a pretty miserable experience. And one thing I'll touch on later is that this hasn't actually changed at all. there are still no good quantitative analysis for doing very, very good tracking when you get situations where cells interact a lot with each other. So what you can do when you do this type of analysis is now we get a full spectrum of what's going on. So not only do we know what our end populations were and our beginning populations were, we actually have temporal information and lineage trees on top of that. So we can track one single founder cell and look at what all the daughters did and look at all the times and how they correlate between them and what they will do. So you can see already that this is a very powerful technique for dissecting out everything that would happen. And this got taken up a notch, and this is what I did with Sarah Russell. I had nothing to do with the development of this technique. This was really Sarah herself with Daniel Day over at Swinburne. And they developed this way of using, making essentially molds so they could put in plastic PDMS, and then they could make little paddocks to then corral our cells and keep them in one area at the same time. So what this essentially did is it said we could do the same experiments, except now we could do them a lot easier. And what you can see now is that the cells are corralled and kept in the same spot, and although I did this experiment, by then all the initial biology was done by Jane Oliara at the Peter Mac, and Kim Pham, who was at the Peter Mac as well, was here at the time and is now here. So you could now say, all right, on top of all the information we have about growth and cell death and what everything's doing, now we can actually measure the lineages of what the cells would do. So now what we do is we turn to fluorescent reporters, and this is something that I'm going to touch on a lot during the talk. And using these fluorescent reporters, we can now go through and then track how we get all sorts of different fate. So the one that I'm going to show for you here, when I get it to work on the screen, is now tracing cells where we have a marker of differentiation, which is here, BLIMP1 for B cells, where all the cells that decide that they want to secrete antibody will become that GFP-positive cell. So now on top of everything that we had before, we've got cell fate, and that's now shown here where we can do another little trick where we can just put antibody into culture on top of that. So that was very good, but we get into this situation now, we're getting more and more and more complex as we get trickier with our technology. And this was seminal work that was done here by Phil Hodgkin that I had very, very little to do with. But it comes back to this point of saying, well, if we can use time-lapse microscopy and we could look at a couple of things happening at the same time, well, let's just keep on adding on markers so we can start to understand the complexity of these fates more and more. So now what Phil's done is he in the lab in a study led by Mark Dowling and then also driven by Ken Duffy, a statistician, is they could then add on top of that antibodies to label the different fate here for becoming an isotype switch cell as well as an antibody secreting cell. The big finding from this paper is that a lot of this fate regulation is actually stochastic. So you have competing fates, whether the cell wants to differentiate to become an antibody-secreting cell, die, divide, and switch. And this is very similar to all the findings that we have for everything to do with division and death. So you can already see it's an extremely powerful technique. But the thing that was always a criticism, and I think was a very fair criticism at the time, was, well, the holy grail is, does a dish equal this? A lot of people are saying, thankfully not, because we already have too much to do with this man. But what we want to do is we want to say, well, can we transition everything that we find in a dish into what's happening in vivo? The important point here is, can we replicate everything that we'd find in vitro, in vivo, in vitro? Can we replicate all the different stromal cell types? Can we represent different interactions that you might have with different stroma in different places that then generate these different outcomes? So that was the big push about why people wanted to move into intravital imaging, that's imaging cells in vivo in living animals. So what you have now is you can look at, well, we might have single cells, and we can look and see, do they do certain things when they're interacting with blood vessels, or certain things when they're interacting with stromal cells, or structural tissue, whatever that might be. So that might say, if we take what we looked at in the dish where we had all these different fates, do they have to be in a certain place for that to occur? Or if when they move around, does it start to get skewed? So you can start to see the exact reason why we would like to do this is now on top of all the temporal information that we've looked at, we also have the addition of complexity of spatial information on top of that. And of course, in a living animal. So, how can we visualize what makes a living organism tick? There have been a number of devices to help do us. One of the early devices, and I don't know if anybody in the audience knows what this was, created in 1952. Nobody's going to put their hand up to know what it is. Because this is actually an E-meter, which is used by Scientologists such as John Travolta, and it's a way of measuring their different thoughts and things they'll be doing. Now, why I like to highlight this is, all right, this is a wacky idea. There's definitely probably somebody in the audience who knows how it works, but this was created in about 1952. The thing about intravital imaging is it's actually been around since the early 1900s. And I know a lot of people are going to say, what? You always hear these people talking about multi-photon imaging and cutting-edge technology, and it's kind of ironic this is an emerging technologies field, and I'm telling you it happened in the 1900s. But the problem there was, how the hell are we going to show this data? So people could observe it and they would make sketches, but it was almost impossible to take this data and show it to an audience that they could comprehend it. So intravital microscopy in some way, shape, or form has actually been around for many, many years. And this is an example of one being built. And so you always get these ideas. People who do this imaging like to pretend like we're ultra special people and that we have some godlike abilities in the machines. They're very temperamental. They're very, very stable. I saw him kick the crap out of this a number of times and it still works. And why I like to show this is that here you spent about $1 million, 1.2 million on a machine, and that you have, in our case at Imperial College, we had 2 split-cycle air conditioners to make sure the thing would stay cool, and they dripped water out of them, and then they broke, and they never worked. So, you know, when we get to technology, a lot of people like to spend a lot of money on the instrument and then skimp on everything else. All right, so confocal microscopy versus intravital microscopy. It is a complete myth that you need to use multi-photon microscopy to do intravital imaging. There are definitely some advantages, and we'll talk about those. But this is the classic picture that you get shown with confocal microscopy. So here we have Eliza. It's a fluorescence being emitted through just a little cube of fluorophore. People would have seen this a lot. It's one of the most commonly shown images. The thing about a confocal is that it shines a lot of light onto your sample, and it uses a special way of then dissecting out the signal that we'll get back in our objective. This is in comparison to a multi-photon, and we have a zoom up here, where we get just excitation at one tiny spot. So how on earth does this work? So it's really simple. The reason it works on a confocal is that we have this little thing called a pinhole. So what does that mean? The pinhole allows us to say, what is the area that we get the excitation of that light? And let's just look at that focal plane. Let's throw out all the other information. So the reason I showed you that sample before is you can see that a lot of that is getting excited, but we're just collecting a small little part of that information. How that works slightly differently with a 2-photon is we don't have this pinhole at all. In fact, you have ones where you might have a confocal and a multi-photon at the same time. You open that out. And you open that up because we want to let as much light in as possible. And how it works is that in a confocal, you have a continuous beam of photons coming along. But in a 2-photon, we fire them in pulses, femtosecond, with femtosecond differences between them. And what that means is then you get an amalgamation or a collection of a high photon density at your focal plane. So that means we get the excitation in 2 of these pulses of these photons. The chance of them hitting at the same time is very, very high, and then we get excitation. So that's shown here. We have pulses of wavelengths of light that collect here at our focal plane. So why do we do that, and why does multi-photon then let us do some cool, interesting stuff on top of that? So if we come back to this picture and we look at it, and now we know how it works, the most important things are these. We get less phototoxicity, and we need less power input to get that same sample. And because of that as well, we can get more depth. And what that means is that as you're changing these wavelengths of light, we get less scatter, and we can get deeper into our tissue. So that's certainly true. But to get that light out, which is essentially the same thing, that's still a problem, and that doesn't change between the two. The other thing about it is nifty, and I'll touch on this later on, is that all the multi-photon lasers are tunable. So people have often gone in and they would have done some sort of microscopy and turned on the different lasers that you need. Well, with a multi-photon, you just type in what you want and you get it. And the other thing is that we have this thing here, which you would have heard people rant on about, which is SHG, or second harmonic generation. So what is second harmonic generation? This is probably my best way of summing up what second harmonic generation. Some of you may or may not know Billy Mays, but Billy Mays is a guy that if you're a very lonely person sitting awake at 3 o'clock in the morning, will sell you all types of crap that you don't need. But the thing that Billy Mays always does is he throws in an extra set of steak knives with it. And that's the best way of thinking of SHG. SHG is your extra set of steak knives. Whenever you do any sort of your excitation for fluorescence, you can then pull out a resonance signal of tissue. And that is different types of tissue have this resonance that they will vibrate at, that we can then get information of that without the expense of a fluorophore. So here's some studies that we've done recently and that are under review now, where we've taken human lymphatic vessels, and actually what we do is we hook them up to a rig and a pump, and then we can pump different amounts of fluid through them, and then look at how the different structural contents of these actually work to change the flow in and out of different organs. And so what we can pick up is by tuning our multi-photon at the right the right wavelengths is we can actually pick up the collagen fibers that make up the surrounding tissue of these vessels, and also the elastin, which is the important part of the stroma that helps to constrict and make these vessels work properly. And there we have them merged together. But we've done this without the expense of having to make any transgenic animals, about having to use any sort of labeling whatsoever. So that's why you want to think of your SHG as well, because you can get all this information about the structure of your tissue without having to use fluorescence. All right, so cell labeling and lineage tracing tools, and that should have had a slide on there. We'll talk about that later. So the important point now is, all right, I've talked to you about this technology and why we want to use it. The things that we want to do on top is we want to use SHG on top of any sort of fluorophore to label the different cells that we want to look at. I'm really unsure, Alex, why this isn't working. I'll just try and bring it up one more time. So obviously, if we're pumping light into our samples, there we go, now it's back. Great. So here's what I wanted to show you before. So we've now got the system working, we can excite everything. Well, what do we want to do with our fluorophores? We have many different fluorophores at our use. So the thing now is we can use these different fluorophores to label different parts of our tissue, whatever they might be. So the great thing about it now, with all the different types of technology, is that we can image lots and lots of different types of fluorescence all at the same time. So here we have an example, and this is looking at bone marrow, and I'll show you later on. Now we're picking up the bone tissue with this SHD that I've talked about. We pick up leukemia cells labeled with YFP, osteoblasts with CFP, vasculature with a Cy5 dextran that we inject in, and vessels with GFP. So by combining all these things together and using multi-photon microscopy, what you can actually do is label all sorts of components within the tissue. So what we're doing here is actually 5-color simultaneous imaging to look at, if we look at an entire piece of tissue, when do we have all these cells present? When do we have all these cells absent? When do we have any combination of them to look? On top of that, though, the important thing that a lot of people are doing now is actually doing lineage tracing. So if you've gone to a talk by Charles in here, he uses lentiviral barcoding to do the types of experiments that I was alluding to right at the beginning. So in those experiments, what you do is you put in a barcode, you do your experiment, you get to the end, you pull it all out, you'd sequence, and then you can go through and look, well, where did the cells come from? Because they're all barcoded if they follow the same lineage with the same barcode. But the thing about that is, again, we're checking out our spatial information and we're checking out our temporal information. So the two big developments that have enabled to now do this with live cell imaging, and I'll touch on this later, is these two systems, which are basically the gold standards in the field. And they are the confetti system and the rainbow system. And how this confetti system works is it's a series of different fluorescent proteins that have different LOX-P sites in them that are recombined in different orders. So essentially what you get now is a stochastic outcome where cells are labeled with one of these 4 fluorescent proteins. A lot of seminal work in this field has been done by Jaco van Reenen, and I'll reference him again later. But you can see now if you begin with a population of cells very early on in tumorigenesis, and this is an example of looking at mammary carcinoma growth, we have 3 distinct populations, a red one, a green one, and a blue one. And what you can do if you look over time is actually look at how these clones compete and then dominate. But on top of that, you can do something that's a little bit cooler, is instead of having one set of these cassettes at the same time, you can then have 3 instead. And what's the point about that? Well, if you have 3 instead, then you build up your number of colors that you can have. And that's what's shown here for this rainbow mouse. 3 sets of confetti constructs in a row, and this gives you basically this huge spectrum of colors that you can now read out. And that led to the generation of some extremely striking images, and this is an example of one of those. So when this came out, basically everybody in the field then wanted to do confetti imaging and then also wanted to do rainbow imaging. But you can see it gives us everything that we wanted to do with perhaps not the same resolution as any of those barcoding experiments, except we know where these cells are and where they are over time. So this is extremely pertinent for all the honors students who will be thinking about potentially buying a house. It's all about location, 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 all right? Maybe I shouldn't have brought that up. So there's 2 different situations that I'm going to talk about today, and they are exposing tissue and then they are using windows. So this was how everything was done very early on. We'd look at organs that were very superficial and easy to access, and examples of those are things such as the skin and the ear as a model for different stem cells, and also the skin itself and different infection models. But also externalized organs. So these were cases with skin flaps or organs that are pretty superficial. So they're things like lymph nodes for imaging and immunology, but also subcutaneous tumors that can be put inside a mouse. Then the skin flap can be exposed as long as you don't damage the vasculature and image very well. But where we're moving to now, and I'll touch on this later, is the problem about this is that the more and more that you sew up and open up and sew up and open up a mouse, you do induce some sort of artifacts for vascular damage and things like that, probably to the extent that we don't know how much it's changing our system. And also vets don't like you very much for doing it. So the thing that you want to do is you want to develop these cases where we have now windows, and you might be looking at these thinking, do they really happen? And yes, they really happen. The mice tolerate them very well. And again, if you look through the literature, you can see many examples of these dorsal skin fold chambers where you have the skin pulled up that can then be re-imaged over and over again for weeks or a month, but also these abdominal and mammary windows that allow us to do the same thing. And one I'll talk about today is this development of cranial windows, of which there's been many different formats. So the thing with this is if you design your experiment correctly, it's very easy to now come back and say, over weeks, months, how does this tissue develop and how can we follow what happens? So I'm going to go through now and touch on a number of the types of studies that have been done in these tissues and why they're important, and especially people that have changed how I've looked at the field over the years and influenced a lot of how I design my experiments and perform my science in general. So a lot of beautiful work has been done by Valentina Greco, who's at Yale, and what Valentina does is she uses the air as a model. The air, now I have a New Zealand accent and my wife can never understand what I say when I say air, but I mean this part of your body, not the stuff around you. So the air is a wonderful model for looking at actually how stem cells work in the hair follicle. And that's what we're looking at here. Here we have a 3D section now moving through these distinct follicles. These are now imaged from the top, and this is going through the X and Y plane. And it's a beautiful system because you can manipulate it very well. You can come back to the same follicles with pretty good resolution over time. And what Valentina did is define the stem cells that make up this follicle. And define where they move, and when you have carcinogenesis in there, well, what are the cells that get affected and how they develop tumors. So you can see already we can get this wealth of information about how these structures move over time and how they're organized, and especially here with the addition, and Valentina is now doing a lot of work with confetti imaging, is to say, well, what are the lineage, what are the distinct parts of the different parts of the follicle that then form these stem cell crypts. This is an example of work that's actually done here with Andrew Liu, where you say, well, what about something that's been done for many, many years? And immunologists in the audience here would know that a long time ago, people played around with transplanting a lot of different types of tissue to a lot of different places in mice. One of the ones that ended up being extremely good was the kidney. You could take a kidney and graft the thymus under there, kidney and graft islets of the pancreas under there, and they'll become vascularized and function normally. So this, again, provides a very easy to access piece of tissue because the kidney can be exposed very well. You can find these grafts because superficially they're easy to see, and they're types of analysis then that you can do. And this is moving through that graft. And the reason that I've paused it here is to say, all right, this is obviously on a curved surface, and if you don't believe me that we're looking at a graft, actually, here's the autofluorescence or the resonance signal that's actually coming out from the glomerula of the kidney. But now what you can do is you can manipulate these T cells in a response against an islet to start to ask, well, what's the difference between the state of them? Are they resting? Are they red? Are they active? Are they green in these Fuki label mice? So again, very, very easy. And to do this with thymus and other organs is turning out to be extremely exciting as well. The one that's being done probably the most is looking at spleen and lymph nodes. And I don't say that because I'm an immunologist and I'm preference towards it, but because a lot of the first studies in it were really driven by understanding the dynamics of the different T cell and B cell responses. The person that's probably one of the famous, along with Ron Germain, is Jason's sister, who's an Australian who lives in California. And again, this is just an example now looking at an exposed lymph node, because anybody that's dissected a mouse, you know there are so many superficial nodes that we can get to. The other thing about them is we can manipulate the tissue around those nodes and stimulate them and activate the immune cells in them in a very, very easy way. So what we have now is the collagen. So this is the example of the SHG. So all it gives you is this little line here, so it's not particularly exciting. But when you're looking down a microscope and you don't know where on earth you are in that organ, it ends up being your best friend. And now we have labeled naive B and T cells in cyan, and the different germinal center B cells in blue. And what you can do is label a number of different immune cell subsets that actually will just take up a dye that you'll inject in. So these are things like macrophages, dendritic cells, and start to look at the kinetics and how these different cells will interact with each other over space and time. And again, this is the first time this sort of analysis was done by Jason and Ron, and it completely influenced how not just our in vivo experiments were, but also how our in vitro experiments. How did T cells and B cells interact with the cells that stimulated them and kicked off these immune responses, as well as the cells that they were looking to kill? And then this is something that's very close to my heart and something that we'll move on to in a second, is looking at bone marrow. This is in the calvarium, one of these calvarium windows I've talked about before. And we have an instance now that we're moving through the section of the bone marrow in the head. And then here is a 9-hour time-lapse movie of just one of these spots and looking at what's happening. So what you have now is labeled all the hematopoietic cells in the bone marrow that make blood, that make immune cells. You have blood vessels all through that label by injecting in a dye. And we can pick up resonance of the bone as well to construct and look at where that tissue is, as well as these green bone-making cells. And I'll talk about why they're important as well. But the nice thing about this is you can develop it and say, before I've been showing you little snapshots of areas, well, if you design your experiments right, you can actually pick up all the spatial information of an entire organ at one time. So this is all the marrow that you'd find in the calvarium, all imaged, but we still have this resolution that if we want to go and do a time lapse of about 10 to 14 different areas, that's still possible. So there's many different applications for this, and this is something that is being expanded on a lot at wehi, and I'm just going to show you a couple of really quick examples. This is a beautiful one that's been done recently, and this is being done with Sameer's lab, and I've written Sameer Towdy's lab because a lot of people have been involved in developing this system. And you can see actually there's so much data on here that actually the computer's struggling to keep up with it. But what we have now is looking at megakaryocytes and blood vessels. The blood vessels are labeled in red, and because the megakaryocytes are secreting out lots and lots of platelets and proplatelets, actually what you end up is that all the vasculature ends up kind of colored yellow. And what we have is these spots now where we can find actually megakaryocytes which connect to these blood vessels and actually secrete out these proplatelets. So it starts to show you that we might be looking at the context of leukemia or immunology when we start off with, but the bone marrow is such an important organ for the function of so many different cells, whether it's cancer metastasis, whether it's just making of these blood, making of these immune cells, we can do it very, very well. And this is the one that I'll talk about soon, is now looking at leukemia cells in red and how they move and interact and how they're spatially distributed through the bone marrow. And I've just shown you a whole bunch of great studies looking at how these cells move and interact with the tissue. So you look at this and you think, oh, that's probably not that cool. But when you work on something that you've stared at for years and years and years, and you go and you kill an animal at the end because it's suffering from leukemia, and you mash up those cells and you take them and you look at a population where you don't have them. But here you can actually look at these cells and stare at them and say, well, what are they actually doing in any space and time and in any situation? It informs your experiment so much to know about how these cells function in their context. It's not funny. And actually you can see leukemia cells undergoing division and things like that in these sorts of studies. The other thing that you can do, and this is a trick that we're using a lot now, but we're not the only people doing it, is to say, all right, all the studies I've shown you so far have been looking at one organ and one animal. But the reality is, and especially when we look at cancer models, is that you don't just have development cancer in one organ. Often they'll start at a primary site and then they'll spread. So if you're clever again about how you design your experiments, you're not just restricted to one organ. And this is an example now of doing bone marrow imaging, which I'll talk about in a second. But at the same time, on the same mouse, when you're finished, and we know that we might have development of a metastasis, or else there's a certain stage that we want to look at, we can actually then also look at the spleen as well. So these are tiles of lots of images stacked together so that we can get an idea about this at a larger organ, so you can, larger level, and probably at an organ-wide level. So the scale here is about 500 micrometers, so it's relatively large. So now what you can do is you can manipulate and treat the cancer that might be fully established in the bone marrow, but also look at these peripheral sites and say, well, how are these cells interacting to cause metastasis? Are they evading chemotherapy by going to different areas of the body? What's the damage to this as well? So this is something that really is what we need to focus on in the future, and a lot of people are doing, is saying, let's not just do one organ, let's have multiple windows on these animals so that we can look at the process throughout the mouse. But I've talked a lot about mice, and I know there's certainly one zebrafish biologist in the audience, because zebrafish are absolutely amazing to do this now on an organism-wide level. And one of the most critical developments of this is, here's a wild-type zebrafish, but there was a mutant developed, which are these Casper now, where you can just see them looking at them very superficially, that they're almost completely see-through. So what this means now is we don't just look at this one of these organ levels, we can look at the entire fish. And this movie, I'll make sure it plays. And this is an example now looking at an entire zebrafish, following on and looking at the development of these bacteria that are growing inside the zebrafish. So you can also, they have a lot of the different immune cells, and they have a lot of different tumor models that we can put into zebrafish and actually look at how they work. And you see it's very easy to see how they grow out in these zebrafish, but also you can manipulate the immune system as well. So you can look at how they interact and how they control infection. So it's not just all about mice. On top of that as well, as I said, they're good for immunology and cancer, but they're also an excellent screening tool, because unlike mice, we can have the ability to create many, many more of them when we create generations of different screens, whether that might be a hairpin screen or a CRISPR screen, and then we can image these with very high resolution and know what's going on. Okay, so do experiments have to have a multiphoton to do them? And the answer, which I alluded to earlier, is no. Confocal sometimes provides more colors. So if you do not need to go that deep into your tissue, you can always grab it and you can always put it in a confocal and you can look. You might not get the penetration that you need, but this belief that you need a multiphoton to do this is not true. And we know that from the fact that people were looking at early blood vessel development and how red blood cells flow through the vasculature in sea urchin in 1912. And that's really interesting if you want to go and look at that paper. But what tends to be very good, and what we've used a lot in the past, and I'll point out when we start to get this so you can see directly what I mean about the quality of this data, is that combination systems with multiphotons have actually been brilliant. So what this means is that if you have a multiphoton laser, and if you block off all of the noise that's associated with that multiphoton laser, and then have a dichroic separating your two signals, you can have your confocal turned on at the same time, and you can collect all your information. So what that means now is you've tacked on a multiphoton with a normal confocal system, and you can get more and more and more colors. Now, this has started to go up a notch recently, and I'll touch on this, is that now people like Olympus have said, well, why do we have to have just one multiphoton laser? Why can't we have two? And it's reminiscent of the situation for the 90s. And I know a lot of the honors students who were probably babies in the 90s and not shaving. But we had the situation, we had a single razor, and then Mark III, Gillette came out and they put 3 on there, and then they said, well, let's put 5 on there, and it just went further and further out of control. And this was the suggestion, that we get to the point where we're going to have 22 blades or something like that. But in the case of microscopy, that's actually not too ridiculous an idea. So we have a system here, and a lot of people have it, where we have 2 multiphotons. And what that allows us to do is get excitation at different levels at the same time, use the detectors that we have set up, because obviously at some situations we get the premium excitation of our fluorophore, and as we drift away from it, we might get a little bit of signal and more for another one. But if we can combine these together, what we can actually do is unmix and get more and more and more colors. So this is the type of situation that we're in now, that we have multiphotons with 2, so we have multi-multiphotons. But it's not unreasonable to think that the future technology will look at more and more, and perhaps not 22. What I should say is that the rate limiting thing on this is probably going to be the cost of the multiphotons themselves, which are not cheap. Okay, so the cases there is traditional labs also, in some cases, and I will touch on this at the end, are reverting away from multiphoton technology. And why would you revert away from multiphoton technology when I've just told you it's so brilliant in some situations? The thing there is sometimes people need speed, and sometimes people don't need depth. So what some people are now doing is they will have a traditional confocal spinning disk system, which was developed a long time ago and always had the advantage of it's extremely fast when we collect data. Also, because we don't submit our samples to much light, we don't have much phototoxicity or bleaching, so we don't lose that over time. So when you're looking at very superficial tissues, so this might be exposed lungs or things like skin, when you want to see something happen extremely quickly, some of the labs that are these traditional heavy hitters in intravital microscopy actually aren't using multiphoton lasers whatsoever. So if you take away one thing from this lecture, it's that you don't need a multiphoton to do intravital microscopy. So what are the futures of this technology? So the big things that are happening now are whole organ data sets, multi-day imaging. And that's the stuff that I alluded to earlier with Well, we get a situation where we can image a mouse for, say, 3 to 14 hours to understand what's going on. But we know from all of our disease animal models that that's not the rate that things happen at. They actually happen over weeks and up to a month and even longer in most of them. So the big push has been to say, well, let's do multi-day imaging and let's also collect whole organ data sets instead of just focusing on these little parts. When you do microscopy, and anybody that's looked at slides, is that you get brought to an area of your attention that might be particularly striking, but that's not a good way to do your science. And it's led to a lot of terrible conclusions, especially in immunology, about how you would do things. You might take out 20 cells, you might take a whole bunch of cells and look at the 20 that do the thing that you're looking at, and then you say, well, look, this is happening most of the time because the ones I wanted to look at did it. And I'm not going to touch on why that is, and I'm not going to touch on the certain studies, but it's a big problem. So if we can look at a whole organ at the same time, then we're drawn away from this bias as long as we do our analysis correctly. So I'm going to start now referring to some of the studies that I've done and set up, and these were set up at Imperial College in Christina Lo Celso's lab. And Mark Scott, who's in the CDI here, was one of the people that was key in developing the early parts of this technology. And what we do here is we take a calvarium window that's inserted onto the top of the mouse surgically, and what it essentially sits like is a backwards baseball cap. It has these small lock and key receivers here so that if you build the correct part that will receive it on the microscope, what it actually means is that you can lock that mouse in and come back and re-image it at the same place again and again and again. Also, because of the design of this, you can seal it up with different material to protect the calvarium and also bandage it, which is what keeps it safe and keeps your tissue integrity. So the types of things you would do is you could image the bone marrow in there by a combination, and a lot of the stuff I show you will be a combination of confocal and 2-photon microscopy, and look at things like single hematopoietic stem cells and how they fall inside the vasculature. So here is an example of how that looks. This mouse is not alive. You can tell that because it's very limp and its anesthesia it has no anesthesia, and yet it's not trying to run away. But what we can do is, here's the example of the rig that will sit on that, and you can design any type of custom pole or receiver here to look at any organ of interest. And this is just an example of the types of things that we would get out of that. And this is actually a movie looking at Pu1 YFP bone marrow. So what we have here is we have these little colonies inside the bone marrow of the development of these granulocytes, which are then feeding into the blood vessels, which are in the dark here. One thing I should point out is, usually if you go and do microscopy and you have a slide, you shine a light through that slide. It's pretty hard to do that with an entire mouse, especially when you've put a big piece of metal in the way. So, of course, you need this external source of light when you're trying to set all this up if you actually want to see anything coming through this objective. So the thing that we wanted to do is we wanted to look at acute lymphoblastic leukemia. We wanted to do this for two reasons. And the first was, coming back to those studies I showed you earlier, we wanted to know, well, if we set this up right, we should be able to understand the prelim data for understanding how polyclonal cells would behave by using a monoclonal cancer cell. So what we're essentially going to do is we're going to ask, when we look inside the bone marrow, if we look at different parts of the organ, do these cells behave differently? The key question that we wanted to look at, and the thing I wanted to focus on today, is to say, we have this problem with ALLs, the initial cure rates are really, really good, but 25% of children and 40% of adults relapse with disease. This disease is refractory to the initial treatment and more aggressive. And the theory was, well, look, there must be cells that are hiding in the bone marrow. They're the ones that reseed disease. And the reason was for that was, when you look at the bone marrow after one of these patients has been treated, the number of cells, minimal residual disease, in the bone marrow will predict with pretty good correlation when this patient will relapse and if they'll relapse. So what I'm essentially saying is, when you have your first treatment, if you don't do a good job of getting rid of this disease, the likelihood that you'll relapse is much higher, and the time at which you relapse is much earlier. So the theory was this. Oh, this crashes again. The theory was this, that when you looked inside the bone marrow, that you would have distinct areas that would then seed and help these leukemia cells behave. And much of this was spawned by the hematopoietic stem cell biology field, where they essentially looked inside mice and said, all right, and I'll get to the data in a second, they said, whenever we look inside these mice, we find one place where we always find the cell of interest. Therefore, ergo, this stromal cell type must be controlling how those cells behave. And the players were blood vessels here and osteoblasts here. Osteoblasts you find lining bone and making bone, and they were always associated with keeping stem cells asleep. So the theory in leukemia was that, well, when we treated with chemotherapy, that some cells would move to the osteoblasts, they would then go to sleep, and because we're using a lot of things that target cell cycle inhibitors and things like that, that's why your cells would evade chemotherapy. And perhaps if you moved into a vascular niche, which was associated where blood cells would be more proliferative, that's where all the cells that were sensitive to the therapy would behave. So the questions were, well, how do they populate the bone marrow? How do they hijack it to survive leukemia? And actually, how do they screw it up themselves? So here's the data that I showed you before now with the imaging window where you collect everything in 3 dimensions. And the point is, here's all the marrow in that mouse. And because we've collected it in this way, for every data set, we can always come back to that and have the resolution to look at it at the single cell level. And here's the type of simple experiments we can do is we can come back over a week and re-image one area of vasculature, which has these green leukemia cells sitting in it, and start to ask the question, well, what happens over time? So everything I'm going to show you from now on has blue for the blood vessels, red for the leukemia cells, and then green for some of the stromal tissue, and then the bone will be in grey. So you can do these types of cool studies, and this is striking, and I like to show it. And I like to show it because it shows you how tiny little pockets of leukemia cells can get out of control in a very long time, in a very short amount of time. So it takes us about 8 to 10 days to find single cells sitting in the bone marrow, and then after about 15 days, we get these little foci developing, and then suddenly, at 22 days, this is what you get. Between 18 to 22 days, you go from an area of a couple of little pockets of these leukemia cells to fully infiltrated. And we have time points along the way as well. But you see the power now that you get to understand the kinetics of your disease and how it's interacting in there. So when you start to do the question, then you say, well, what do they look like when we videotape an area over an amount of time? How do these cells look? What do they interact with? These are the types of fundamental observations that intravital microscopy lets you do. So this is one I love to show, and this is actually a single leukemia cell just moving along between this space between blood vessels and these osteoblasts. And what I want to point out is this is all confocal data. None of this is multi-photon whatsoever, and it works really, really well. So what we like to do is we like to create these 3D images, and we call them Google Earth-like images, so that we can then couple this as quantitative analysis to start to ask the question, where do these cells go? And this is to directly address the screw-up in the field about how these cells would behave. The point being is that if you look at this, what you can tell is that the blue are the blood vessels and the green are the osteoblasts. They're ridiculously prevalent. So if you do a study where you don't have the right type of control in there, and you say, well, I'm going to measure what these cells are close to, and we say, well, look, they're close to blood vessels, so the blood vessels must be controlling what they do. Also, these osteoblasts are far more prevalent than most cell types in the bone marrow. So you say, well, look, they're close to osteoblasts a lot, therefore the osteoblasts must control what they do. And you might think that that's ridiculous, but that's actually what a lot of the conclusions have been for many, many years that are now looking like they're not true. So what I want to highlight is that the field of image analysis is really at this crossroads now, where it's starting to become ridiculously important to us. It's almost at the stage of bioinformatics with the explosion of genomic data in the early 2000s, where basically we couldn't do anything we wanted to do without a good bioinformatician. And before that, they were at the stage where they were almost certainly unappreciated. And now we're at the same stage with image analysis people that have these skills to dissect out and pull out quantitative data to address these problems that I've just told you about. If you cannot look at your data properly, we don't have things like FlowJo for intravital imaging data. You cannot perform it properly. So coming back to that data I showed you before, well, here are the controls for those. And what we did is we said, look, if you simulate cells that have no inherent ability to go anywhere, if you do them randomly or uniformly and then overlay them on your data, that's really the only correct way to do this, because there doesn't exist a proper positive and negative control for this whatsoever. And what I want to say is when you do this type of analysis, you don't find any difference in where any of those leukemia cells want to go whatsoever, and I'll show that in a second. So the big thing is, what happens when you do and don't have chemotherapy? Let's use the multi-day imaging now to say where are these cells going and how are they behaving. So here's the first experiment we ever did. I always show this. I love showing it because I thought it was a failure. It was a Friday night, and I thought I'm wasting my time. Here is the mouse 2 days later after dexamethasone, which is a standard treatment. And what you can see is it's done a very good job of controlling this leukemia in red, and we can find little single dots of surviving cells. And when we did that analysis, we could pull them out here and see them very well. And we could overlay the simulated data to look at different stroma and ask the question, is there a certain area that's controlling these cells, keeping them asleep, selecting out for the ones that didn't want to have anything to do with that chemotherapy? And the answer is no. But what we noticed is that if you videotape them and looked over 3 hours and said, what are these cells doing? Well, we actually got the complete opposite result, where they weren't homing to any area whatsoever. They're moving rapidly around the mouse. And all these timescales for the other data for leukemia I showed you earlier with the single cell tracking are the same. And what you can see is actually they're moving faster. So much so that when we do this with ALLs from humans that are xenotransplanted into mice, and we label now with antibody labeling, we get the same result. But as we add in more and more different types of chemotherapy, we thought that this theory was going to fall apart. And actually, we've got the complete opposite results. And now we're not doing just dexamethasone, we do vincristine as well, and dexamethasone, vincristine, and allosparaginase. And actually what happens is, as you add in more and more chemotherapy, it works better as you would expect, but actually the cells move faster and faster and faster, despite even the fact we have these G0 cells that aren't in cell cycle. We still have them moving around, zooming around the bottle. We haven't selected for some sort of chemotherapy-resistant leukemic stem cell population that is quiescent and stuck in one area whatsoever. So the last question I want to say is, well, does anything actually happen between these cells whatsoever? And the striking observation was, this is that type of data I showed you earlier, where we have the normal cells interacting with the green. And these are the osteoblasts, and now we have it in this leukemic situation where actually the leukemia rots away and gets rid of these cells completely. And this actually, about a month before we published our data, somebody came out and said in human biopsies they were seeing the same thing. But what, again, the intravital microscopy shows you is that the speed in which this happens, at over a 9-hour period, we go from having pretty healthy cells, so they're pretty much rotted away, and if we don't treat the mouse whatsoever, this is what we get. Okay. So I'm going to flick over the last few slides very, very quickly, but just to say that we have many, many different tools to look at other different parts of the components of the bone marrow, now looking at the blood vessels and how they work. And this is shown even more beautifully here with this this is actually confocal data on top of multi-photon data. So again, highlighting that fact you don't need a multi-photon to collect this information. And when we look at AMLs, we actually see the completely different thing that we saw for the ALLs, and that we don't see remodeling of the blood vessels at all in the ALLs, but with the AMLs, we actually see complete remodeling of the blood vessels, but not the osteoblasts. And actually, to the point where we start to look at those, you can see the blood vessels are shaking around, and also the endothelium starts to form these cilia actually to interact and remodel. So this is actually packed full of leukemia around here. So these are the types of things that actually, when you visualize your data using intravital microscopy, you can pull apart. Okay, the last thing I wanted to say is photoconversion is actually the final future part. And that is a system where now you can bleach and change the color of populations of cells depending on different constructs that you have there. And what this means is that now we're not just imaging a stagnant population of cells and saying, well, we've labeled them, we know what we're doing. We can change these cells from being all green to red with just simply a zap of the multi-photon laser and start to count and quantify and see how they change over time. So again, incorporating this day 3, 4, 5 imaging using these different types of windows, and these are mammary windows shown here, and looking at how these cells actually wrap around and they probably move into the vasculature and metastasize as well. The final frontier is actually incorporating this with photoactivatable Cree systems to turn on and off genes in its specific places. There's a lot of work being done on this. Some people are doing it in zebrafish and doing it pretty well, and some people are doing it in mice. But once we have that, that will then say we can label those surviving leukemia cells and see where they go and what they do and what they were so special, what was so special about them. So the final thing I want to say is that it's on limitations. In intravital imaging, there's always a trade-off. And of course, here's a guy, well, he's not a guy, he's Hulk. But you never skip leg day. So if you want to get better resolution, you are going to lose speed. If you want more and more colors, you are going to lose speed. And everything you do in that intravital imaging is a trade-off. You might want to image more and more sites within that bone marrow. But then again, you're not going to be, you have to trade off information all the time. So this is the biggest thing about it, is we're always after speed. We want speed, speed, speed. We want to collect as much data as we can. We want as much resolution, but none of these things come out with the trade-off. If you have twice as much the resolution, it takes twice as long to collect, which means half as many positions, which means then you have to double your amount of mice that you have to do to get statistical significance. So that is the hugest thing about it. And you sit there and you get to the point where you might actually have one little last image that you want to collect, and you have to trim little bits and bobs out of all your Z-stacks just to squeeze out that last part. And that's the thing that I sit there doing, and it drives me absolutely crazy. So I'm going to stop there and take questions. I just want to thank all the people involved in the work. My lab, who are here, a lot of the setup for the intravital was done in Christina's lab. We get a massive amount of help here with the CDI, and there are a few talks that are going to be here on imaging, and I suggest if you want to do this, talk to the lab head there, Kelly and her team, who are absolutely brilliant. And then we've had lots of help along the way with other people who have provided leukemia samples and the setup of all the initial cell tracking here by Phil Hodgkin's lab. So I'll take questions.
Good job. Thanks, Ed. I think just like a lot of other talks we've heard this year, I couldn't agree with your last point more. I think it's really important, and I think your talk is another prime example of how we need a cross-disciplinary approach in biology, and we should be talking to physicists, and I think, and how this is how we're going to drive innovation, both from the 3D printing of your little bits on the microscopes, the anaesthetics, the fluorophores, the colours we're using. It's so much to it. I'm sure you're all aware attendance and asking questions is an academic hurdle for some of our students, so if I could invite some of those first, and then I'll open it up to the rest of the audience.
In terms of the data collection and analysis, how much of that is automated? How much are you doing yourself?
Yeah, that's a great question. So a lot of it is done yourself. The biggest problem is the manual tracking, because if you get a case where you have 2 cells come and then they do that and they go off, any tracking algorithm will track that, but they cannot be certain. So the way that you want to interact with that data is you want to say if you can't be certain, you have to ditch it. If you leave any tracking to do that automated, it's a disaster. And we know that from the in vitro tracking and the in vivo tracking. And there have been so many algorithms that have been designed since, and I actually think there's a score somewhere from the manually tracked data that I generated where people, I think it's called the ED score, where people will actually track and look at how well their automated algorithm does. And the answer is there's a lot of conflict there where it doesn't work. Where it does work really well is the segmentation, the analysis of that 3-dimensional data. And that, it works extremely well because you're thresholding on colors that are very easy to dissect. The green is the green, the red is the red. In most of those cases, unless you get the a couple of little dots of pixels, if you can resolve out a structure and know it must be at least 20 pixels large to be a cell, so everything underneath that is junk, and it can't be more than 1,000 pixels, and it must be green, then you can do that type of analysis. We did that in Definiens, which was the most painful thing in my entire life, because to analyze 10 cells would take about 3 or 4 days of computational power when we first did it, but it had to be done that way to take out the bias. So it's a horses for courses. There are a lot of automated systems that are good. Lockie here is providing massive help in developing a lot of custom things and doing a great job of it, but there are also some that have to be done manually as well.
The glass is over.
So a lot of your cell tracking was in vitro. Would those same techniques work in vivo?
They do, and it comes back to what I was saying before. The principle is thresholding, right? If something's black on a white background, you can track it, because all you do is you say, well, everything that's black will pseudo-color in the thing that we want to follow. So that gets very easy. And that works for all the fluorescence on top of that. So you get fooled into looking at these pictures that have got different colors in there, but they don't. They're black or they're white, right? So if you have good enough resolution to see that, then that is fine. So that works exactly the same for intravital imaging. You've just got an extra dimension. So yes, there are some situations where some of the tracking where I showed you where you only have 1 cell, all the automated tracking works brilliant, and it's the same thing. It's either black or it's white, right? You know its size and you know it goes somewhere. But if you have things that move really quickly, you can never be certain where they are, and that's when you start to get into trouble.
Aaron, do students have nominated? Anyone else who's nominated for today? Okay.
I'm just wondering about the ethical considerations of attaching windows to device. Is it difficult to get approval for that? Or?
Yeah, so we spent a hell of a lot of time going through that, and we did the United Kingdom, and when you do it in the United Kingdom, you learn that how we do animal work here in Australia is very, very loosely regulated.
More loosely.
Very loosely regulated. If you want to do animal procedures in the United Kingdom, you have to do a 4-day course, you then have to pass exams, you then get considered for months on end, and then they will award you a license, and then things are very specific. So actually, there's 2 ways to look at this. It needs to be done right. That area that we re-image is poorly vascularized, and that means that as far as pain levels go, it's very, very low. So the amount of, they're kept on analgesia for the entirety of that project through given painkillers that are in jelly that they eat, but actually they've now said that they don't need those painkillers. the thing about it is we can reduce the number of animals that we use significantly. So something where you might have to take 5, 10 mice for a cohort and do that again and again and again, by doing the same mice again and again and monitoring them correctly and caring for them because they are important, then that is actually the way to do it. So if you do the experiments right, it is amazing how well they deal with each other. They live with their litter mates. They do not care at all whatsoever. They do not behave differently. As soon as that happens, you have to kill your mice, but they're amazing animals. But you have to do your experiments respectfully.
Aaron.
So how do you design a good antibody like staining panel for this? Because like if you have like to use two photons to excite the antibody, right?
Mm-hmm.
So like how do you like does that affect your resolution if you use like different colors?
So the answer is yeah, it does. And we've done a lot of work with injecting in different antibodies to see how it works. So I showed the stuff in vitro, and that works very, very well. You can label cells for sort of 4 or 5 days and keep on imaging them every 2-3 minutes for 700 milliseconds with an epifluorescence lamp, and they will give you a signal for that entire time with very little degradation. When we go in vivo, of course, you're probably getting less of that fluorophore on those cells at a per cell basis. So we get about 3 hours when we inject in an antibody. And what you tend to find is that PE works better than FITC, works better than APC. And the exact reasons around that, some of them is to do with excitation. Some of it to do is how good your laser is. Sometimes if you're using a confocal laser, your 633 might be slightly weaker than the other one. So it is a consideration, but it works really, really well. And the answer is it often comes back to how much your antigen is expressed. Things like CD45 on immune cells expressed a huge level, but we can label stroma on the FLIC1 endothelial cells in a blood vessel. You can inject in an antibody against it and image it for hours and hours on end, and the overlay between the GFP for your reporter and your antibody are pretty much spot on. But my comment on that would be it should be used as a way when you don't have your transgenic animal and you need to do an experiment quickly.
Student question here.
Cindy?
Open it. Oh, sorry. Who did I miss?
Cindy?
Cindy. Sorry, Cindy. Yeah.
So when you want to introduce many colors, like you mentioned that people actually even use 5 colors, but then how can we be sure that, if, especially when you do more than 1 day imaging, how can we be sure that there will be no effect of the biological condition of the...
Great question. The answer is, when you set up your first system, you do cohorts. You have to do cohorts. So how we did the leukemia imaging is we started a cohort of mice with 3 on day 10, and then on day 12, re-imaged those 3 mice and re-imaged 3 more mice, and then you kept on going. And you do that the first time you do it, so you know any single artifact that you might introduce right at the beginning of your experiment. Because if you go for a week or 2 and you don't look after that bone, and Mark, who is sitting right next to you, will say he knows when it falls apart and your bone looks shocking. You know, but it's the subtle differences. One thing we don't know is there's already people talking about excitation of immune cells through blue lasers and green lasers. It's actually talked about in CAR T-cell therapy in the clinic. So there's some things that maybe we don't exactly know. So the answer is, so you set up your experiments carefully, you make sure that as many of the variables you can control, and as soon as you can't, you stop your experiments and you don't do it like that.
Right, I'll open up to the whole audience now for a final question. Is there any burning questions?
I have to ask one.
Oh, sorry. I'm
Thanks.
sorry, you're not on my
Thanks.
list.
I have to answer it if that makes you feel better.
It's basically the same as that one, but I guess another question I have is how deep can these current techniques go?
Yeah. So the answer is it's very tissue specific. There are some tissues where you can go, so we were doing stuff the other day here where we were going in live tissue, going 250 micron deep and not having a problem whatsoever. So this is why it's all about what organ you're imaging and what you have to take out that's in the way. If you want to graft something under a kidney capsule and image it, you've got a very fine layer in the way. The calvarium bone is extremely thin. If you dissect a mouse and you're like, what does this bone look like? And you have time, just chop it out and look at it. It's almost see-through. So that's why it's so advantageous when we can get so deep. But things like spleens and things like the liver that have packed full of red blood cells, we start to get more and more trouble. But it's all about signal to noise. If your thing is expressed really dimly, then it doesn't matter how thin it is. you're not going to be able to see it. But if it's extremely bright, you can get deeper and deeper. So again, it's always a trade-off of these things.
Thank you. Just remains for us to thank Ed for a great talk.